Okay, it's about 1 o'clock on my computer, so we're going to start this session. Hello, everybody. My name is Miles Daniels. I'm going to be moderating this session today, which is titled Drought Impacts. And before I start, I just want to thank all the presenters for taking their time out of their very busy schedules to come here today to share some of their research. As the title states, we're going to be looking at drought impacts, both at physical systems, biological systems, for components in the upper watershed of the Sacramento River, as well as down in the Delta. There's a very nice, diverse set of talks you have to listen today. And so without any further ado, I will start by inviting Sarah Null, who's an assistant professor from Utah State University, to go ahead and share her slides after I stop sharing mine.
Okay, can everybody see those slides?
Yes, we can see them. Thank you.
Okay, great. My name is Sarah Null, and I'm really excited to be here at IEP Workshop. Too bad it is virtual. It would have been nice to be there in person. Today, I'm going to be talking about some work I've been doing with the Public Policy Institute of California on storing water for the environment. My day job, I'm an associate professor at Utah State University. So environmental water management is not very proactive to drought. That's one of the reasons we're having the session on drought impacts. And there's a lot of reasons for that. So one of them is that most water for the environment doesn't have an actual water right or priority. So it's more of a constraint on other water rights. We operate water in California so tightly that there's not very much room for error. And then we have delayed institutional responses, or another way of saying it that is there's not very many changes to be made in the middle of a drought. We kind of have to just stay the course. And to show that, we can look at this figure here I put on the right. This is water year 2021. So this is last year, critically dry year. The y-axis is average monthly percentiles. And you can see the blue line, this is cumulative reservoir inflow for like the 10 largest reservoirs in California. And you can see how it's low. It starts the year low and it's dropping, but we were still making big reservoir releases. We didn't really fail to incorporate that there wasn't much snowmelt. We didn't really get that anticipated plug of snowmelt coming in. So the green line, the water storage in the reservoirs just continued to fall. So because of that, we had a drought declaration in May. We had a TUC order, or temporary urgency change petition, so that we had a new temperature management plan. Finally, in almost July, post-1914 water rights were curtailed, and then not until about September were pre-1914 water rights curtailed. So we were really on this crash course last year of not having very good tools to manage water for ecosystems and for the environment. So because of that, at PPIC, we've been thinking about one of the things that might be promising is to expand storage options for the environment. So as a little bit more background, we know that droughts are intensifying in California. So this plot here is showing the temperature departure from normal over the last 120 years. And you can see that we start in some pretty cool years, but the last 20 years have been not only dry, but also really warm years. And this makes sense with climate warming. If we were to look at runoff, it's the same kind of departure for normal, except for we're getting drier and drier and drier. With that warming and drying comes more volatility. So we have, sometimes shorter wet seasons and longer dry seasons, or many dry years punctuated by these very wet years. And that's tough to manage. And NOAA really epitomizes that quite as much as Oroville. So here I have a picture of Oroville in 2017 when the spillway was failing. And then last spring, when Oroville was so low, we were struggling to generate hydropower and meet water supply needs. Okay. And so that brings us back to then the idea of reservoirs and how do we manage them for the environment? And we have a paradox here. So on the left plot, I'm trying to show that dams harm freshwater ecosystems. So everywhere in red are places where habitat has been disconnected because of dams. And we've known that dams harden ecosystems for decades, right? That is nothing new. But the newer part is also thinking about sometimes we need dams to provide environmental flows and water quality standards that are regulated, that are mandated, by sometimes BIOPS or FERC licenses or state water board decisions, sometimes water quality, Clean Water Act. And so we really need reservoir storage to be able to achieve these flows and temperature standards. So because of that, at PPIC, we wanted to look at what would it look like to store water either in surface reservoirs or underground to be able to better meet ecosystem objectives. And the idea with this is to really manage environmental water. So thinking about storing water, sometimes carrying over water, so among years and between years, being able to store that water, sometimes trade it or market it, exchange water, basically have all the tools and strategies that other water users have. Again, the idea is to really have tools to be able to manage ecosystem water so that when we get into droughts, we have those tools. The idea is also to move away from the rule-based minimum in-stream flows that are just tough to manage when you get into dry periods. And this idea, I know it sounds kind of extreme and like, oh, this will never happen, but it already is. It's one of the ideas for the Proposition 1 projects, where Prop 1 projects have to have public benefits, and often that's through meeting ecosystem objectives and priorities, and sometimes that has storage involved. So we are taking a three-pronged approach to looking at the idea, exploring the idea of storing water for the environment. One is looking at infrastructure, and that's what I'm going to be doing here today. I'm going to be talking more about the technical modeling side. We also have other folks on our team looking at governance and legal aspects, and I'm not going to be talking about those too much today, although we have a report coming out in about maybe May or early June when we'll have all of this together. And again, the big goal that we're focusing on here is improving ecosystem health through managing stored environmental water. So I'm going to start with really quick how reservoirs work. And this is probably, background for everybody. So this figure here is showing through one water year. The y-axis is reservoir storage. During the winter, we have to draw down reservoirs where we have a flood reserve. Usually at that time in the winter, all of the reservoirs cold water. And I'm thinking here of like large reservoirs like Shasta or Oroville or maybe Folsom. We start to get snow melts in the spring into even early summer. And then we go into irrigation season when the reservoir gets drawn down. And also when we have, warm temperatures, high sun angle, and we start to have that thermal stress, and maintaining and managing the cold water pool becomes really important for large reservoirs. So to model this, we took a really simple approach where we made a water balance model and we paired it with a reservoir temperature model. We didn't try to represent any particular reservoir, just kind of a generic or hypothetical large reservoir that stratifies. We allocated a proportion of inflow for the environment. We used 10 to 40% in different model runs, and then a proportion of storage, 10 to 40% storage in the reservoir for environmental objectives. Environmental objectives were only some of the water demands. We also tried to give, some sense of reality and added in-basin exports, salinity requirements, and refuge requirements. We ignored hydropower and other uses and just looked at water supply. So I'm going to walk us through the environmental objectives so the results make sense. So the first thing we did was what we called environmental base flows, and those are the water needed to meet water quality standards and environmental flow. So that's the water that's required to meet the regulatory standards. And this figure here is showing them. We modeled this monthly for 26 years. And this is showing the blue lines are 10 to 40% of inflows if you just pass that through a reservoir. And then the red lines here are those ecosystem base flows to meet regulatory requirements. You can see that they change among months and between years. And then we had some temperature management because we managed for cold water species in California. And so the idea of temperature management was to provide suitable temperatures for winter run, fall run, and late fall run salmon. And I have tables to back this up. If anyone's interested, I could pass them on a little bit later. And then we wanted to bring in the idea of functional flows. And so here I'm just we modeled just blocks of water with the idea that a reservoir operator, ecosystem manager would actually manage this very different, release these in large pulses to have the greatest environmental benefit, but we just set aside a volume of water for now. So the first big takeaway message is that having carryover storage enables environmental objectives to be met much more often. So here this is showing 3 water years. So water year 2019, a wet year, followed by 2020 and 2021, which were increasingly drier. 2022 is going to be on that same, probably critically dry area. So starting here at the top figure, this is showing we mandated a cold water pool, and we had to do that to be able to meet temperature requirements. And I'll talk more about that in a minute. This also mirrors how water is managed in California, where operators try to end the year. They try to start October 1st with about 1.5 million acre feet of storage. Sometimes in wet years, that's even higher. And excuse me, and then the light blue line here is showing water supply storage for just all other supplies. And the green line is showing environmental storage. So you can see how some water is carried over from a wet year into the drier years. By the end of 2021, it's pretty much all used up. We were just short of water. We weren't short of storage, we were short of water. This next flow is showing the environmental base flows. So those regulatory requirements that we have to meet in California. We could meet all of those. That was not actually the case in 2021, remember. And then the bottom figure is showing the functional flows. And so here I have a spring recession, a winter pulse, and a full pulse. By 2021, we didn't have enough water to meet those functional flows, to meet that more water that we can manage more flexibly. And then of course, if we allocate water and storage to the environment, there would be trade-offs with other users. And so in this figure, I'm showing those trade-offs. So the x-axis is total ecosystem water shortages, and the y-axis is water supplied other users. So, ag, urban, other users, their shortages on the y-axis. This green point here is the utopia point. That's where we want to be ideally. These pink lines here are showing if there's no storage and just passing flows through for the environment. So you can see even with 10% of inflows passed on through the environment, we can't meet a whole lot of environmental objectives and priorities. As we start to add storage, that decreases and we start to shift the shortages over more to other water supply users. And so the idea here is we're not necessarily saying exactly where we should be, but we're trying to explore these trade-offs and understand really, when environmental objectives are met and when water supply objectives would become really just overwhelming. And then one of the really interesting findings was that there were trade-offs between managing flow for functional flows or environmental base flows and maintaining that cold water pool. So this figure here, this top figure in the tan colors, this is showing 10% of flow for the environment pass through, no storage, and then 40% is the darker tan color. And you could see that the cold water pool, these are showing the 12-degree cold water pool volume in a reservoir, and that really starts to get depleted as reservoirs stage drops down and as more stream flows pass through the environment to meet downstream environmental flows or functional flows. So one of the best ways to do this was to maintain or stipulate that a cold water pool is maintained. And our modeling showed that about 1.25 million acre feet or about 1.5 million acre feet is really the gold standard. You get below that and we couldn't meet temperature requirements downstream of dams. And then storing water for the environment is a promising way to adapt to drought intensification. It's not the only way, but it's one that we've been exploring so far because we know that we're going to keep having droughts in the future, probably more frequently than we did in the past. So we know we need some tools to be able to manage them. And here's the rest of my team. This is all, there's lawyers and geomorphologists and ecologists. We're all working on this project at PPIC, and this is my contact information. Thank you.
Great, Sarah. That was really, really interesting. So we'll open it up for any questions that we might have for Sarah. I see. It looks like maybe Deidre has her hand raised.
Yeah, I was wondering what modeling you used for the pass-through flows, the 40% and 10%, to get the conclusions about reservoir storage.
Yeah, we made a water balance model. That's a good question. We made a water balance model. So we took water demands throughout the state and we simplified them and scaled them down to be a single reservoir. And then we paired that with a model called WQRRS, which is water quality for river and reservoir systems. It's a HEC model, and we use that to model temperature.
How would you expect the aggregation to affect the results?
I would expect, so in our modeling, we tried to represent senior water rights holders, junior water rights holders, and environmental water demands. And so I would expect us to generally, be able to quantify trade-offs. I wouldn't expect us to match actual operations where there are like settlement exchange contractors or any very specific water demands and water rights. We didn't try to match water rights and really get into that level of detail.
Thank you.
Yeah.
Next question. Looks like Bruce also has a question.
Hi, thanks. This was great. Enjoyed that. You mentioned Prop 1 and the infrastructure, and the big thing out of Prop 1 is sites reservoir. And there's a lot of talk about the kind of environmental water that you're describing here. Do you have any thoughts, or have you built that into how it would work and how its effects would be?
We have done that. First of all, we've had some conversations with some of the Prop 1 managers, and then we've done some model runs. I didn't talk about them here because of time, but we've done some model runs where we added storage. We didn't specify whether that was surface or underground storage, but you could say, oh, this represents if sites were to be operated wholly or partly for environmental management. And it's really interesting because we could really quantify, especially in wet years when there's not storage capacity in our existing reservoirs, we could quantify the environmental objectives that we could meet from having a reservoir like sites or conjunctive use or better using underground storage. And so, yeah, there are definite environmental benefits to having additional storage.
Thanks.
I have one question while we're waiting for any others. You may have covered this. For the meteorological forcings for your water temperature models, were those based off kind of a historic or more of a forecasted climate change?
They were all historical, and so everything we've done so far is historical. And we did not get into in this modeling effort. We did not get into anticipated climate change effects. Yeah, good question.
Good next steps, perhaps.
Mm-hmm.
Okay, well, I think we can likely move on to the next presenter. Thank you, Sarah, so much. That was really interesting. And I wish we were meeting in person because I'd love to follow up more about all the work you're doing. Thank you so much, though. So next, we have Rosemary Hartman, who has a pre-recorded talk, and she's an environmental program manager with the California Department of Water Resources.
Hi, everyone. My name is Rosemary Hartman, and I'm from the Department of Water Resources, here to continue the discussion of drought with a synthesis project that the IEP synthesis team did, looking at the ecosystem responses to drought in the Delta. We had a huge team looking at everything from hydrology to fish, and they used almost all the IEP data sets we had to see what the situation is in the Delta when we have a drought. I want to acknowledge all of the hard work of all of the IEP people who contributed data to this effort and all of the folks who helped with technical advice, review, and support. So, what exactly do we mean by drought in the Delta? Well, when we have a drought in the Delta, I think we all know about the California basic water cycle. It snows in the winter. That snow accumulates in our mountain snowpack. In the spring, the snow melts, flows downstream into our rivers and into the Delta. When we have high flows, we tend to have higher turbidity, lower temperatures, more native fish, and often more productivity. Everyone's happy. In 2021, things were different. We had less snow, and instead of flowing down the river, a lot of the snow we had evaporated or sank to the soil because it was so hot and dry. This meant that we really had to scramble to figure out what to do. Department of Water Resources and the Bureau of Reclamation had to submit a temporary urgency change petition asking for changes to our water quality standards because we couldn't have enough outflow to meet them. We had to install an emergency drought barrier in West Falls River. Hatcheries changed their production and installed emergency coolers. All sorts of things happened, and we really had to scramble to get it all done. In the Delta, those low flows mean that we have clearer water, higher temperatures, more non-native fish, and harmful algal blooms. So even with all of our attempts to mitigate the situation, it was a bad year. When I say drought, most people think that means less water than usual. But in California, there's not really anything unusual about droughts. In this graph, I show the Sacramento Valley Index, which is the idea of how much water we get on a year-to-year basis. And looking back since the turn of the 20th century, we see that droughts are not particularly unusual. We have dry years all the time. What we decided to do was not just look at a single dry year, but look at when we have an actual extended dry period or drought. Past 6 recent droughts, when we've had good monitoring, were ones where we have multiple below normal, dry or critically dry years in a row. We compared that to what we called wet periods, which is multiple wet or above normal years in a row. And we also have what we call neutral years, which is when it flip-flops between wet and dry. You'll see those classifications used throughout this presentation, so kind of keep that in mind. We did a 2-pronged analysis. We first wanted to look at long-term trends to see if there are commonalities between the droughts we've had in the past, looking at all of the data we've collected since 1975, and comparing droughts to wet periods and neutral periods. We also took a closer look at 2020 and 2021 to see, okay, is this most recent drought similar to past droughts, or is it different? We looked at data sets from across IEP using all the ones that we could find. This is where the hard work of the data utilization work group and all the IEP principal investigators who've published their data with good, robust metadata has really paid off. It was really easy to take these data sets and integrate them and compare them. Well, not really easy. This was a ton of work, but it could be done in a very abbreviated time frame. So big kudos to everyone who worked on it. And I'm going to skip over the details there and get straight to the results. We did a lot of work organizing the data, and this is what we found. Starting off with hydrology. Well, delta outflow really decreases during droughts. No surprise there. I'm going to be showing you a lot of graphs with this basic format. Drought years versus neutral years versus wet periods, which drought is in yellow, neutral is in green, wet is in blue. And then 2020 and 2021 will often be next to them. And 2021 was lower outflow than we've really ever seen before. I do, though, want to point out that we use outflow as calculated by DWR's dayflow model. There are other ways to calculate outflow, and they don't always agree. USGS outflow calculation corresponds pretty well to the dayflow calculation at mid to high levels of outflow, but really low levels of outflow, like during a drought, they don't correspond that well. So we don't even really know how low the outflow was during these drought conditions. Water project exports also change with drought. We tend to have much lower exports during drought years, and 2021 was some of the lowest on record. X2, which is the point where the bottom salinity is at 2 parts per thousand, also shifts, sort of as expected, with less outflow coming in, you have more salt coming the other direction. And 2021 was as high or higher than previous droughts. For water quality, it was hot. It's usually hot during droughts, but 2021 was worse than other droughts. Water is also clear during droughts, higher Secchi depth on average, and the most recent 2 years had some of the highest Secchi depths that we've seen. Salinity also, again, like X2, as freshwater flows decrease, salinity increases. The increase in 2020 and 21 was partially due to that emergency drought barrier and change to water quality standards. How does this change in water quality affect the critters that live in the water? Well, when we look at phytoplankton or chlorophyll, we tend to have the highest chlorophyll actually in the neutral years. It's hard to see in this graph. The neutral is actually slightly higher when dealing with the autocorrelation and differences between regions. On average, neutral years are the highest in chlorophyll, but it's different between regions. So in the South Central Delta, across seasons, you can see that neutral years are usually higher. But in Susanne Bay, the wet years are often the highest. That's the good algae. Well, what about the bad algae? Microcystis is a harmful cyanobacteria that has been seen in the Delta since 1999, and it's measured using a visual index from absent to very high. We've grouped it into absent, low, and high just to make it easier to analyze the data. And we find that the wet periods are much more likely to have no microcystis present, whereas during droughts and even neutral years, you're more likely to have low or high microcystis present. And this is most common in the confluence North Delta and South Central Delta. So they definitely like droughts. This past year in particular was bad in terms of delta-wide microcystis, but we had a particularly bad bloom, which you can see in this yellow-orange color here in July and August in the Central Delta and Franks Tract. This data is from the Flora probe that DWR's environmental monitoring program has in their water quality cruises. Similar thing was found in USGS's water quality cruises. And we could use satellite data put together by SFEI to show the same patterns in Franks Tract in July and August. So that's the bottom of the food chain, highest during intermediate years, but different in different regions. Zooplankton is kind of the next step up the food chain. And we also see some pretty big regional differences, with the South Central region and Susanne Marsh tending to be higher during droughts. Neutral years varied, but it's interesting that confluence, there wasn't a big change with drought. In the South Central region, total zooplankton actually decreased during droughts, whereas in Susanne Bay, it increased during droughts. And we see some differences in total biomass between regions, but a much bigger difference in community composition between regions. If you want more on this, definitely check out Arthur Barros's talk in the lower trophic section. more cladosterins in drier years, especially in the south central delta, more pseudodiaptometh and mysids during the wet years, especially in Susanne. What's driving these patterns that are really different in different years? One factor is invasive clams. So the freshwater clam, Corbicula fluminia, doesn't have a big difference between droughts and wet periods, whereas the brackish water clam, Corbicula amurensis, has a much higher grazing rate during droughts than wet periods. And this tends to live mostly in Susanne Marsh, Susanne Bay, and a little in the confluence. During droughts, it's eating a lot more of that phytoplankton, so you can expect that phytoplankton will be lower in Susanne during droughts. It's not in the south central delta very much, so it's not controlling the biomass as much. But it does move upstream during droughts. Salinity intrudes, and so does Corbicula, particularly when we look at the previous year's Sacramento Valley Index. So it takes a year to shift, but then it shifts upstream with drier years. Now, everyone always wants to go to the fish. What's going on with fish? Salmon, everyone's favorite fish, we calculated the cohort replacement rate, which is the number of adults per juvenile. So for every one baby fish that goes out, how many adults come back. And we found that for fall run, spring run, and winter run, neutral years and wet years had significantly higher cohort replacement rates than droughts. There's a lot of things that happen between when a little baby fish hatches upstream and when the adult fish returns. It's not clear exactly where on that stage this change in the cohort replacement rate occurs, but there's probably higher mortality in the hatching grounds. There is probably lower survival as they outmigrate. There may also be ocean conditions that are involved. Suffice it to say, if you're in a drought year, it's not a good year for salmon. Looking at our other fish that are out there, some of our favorite pelagic fish, threadfin shad, there's a trend towards higher abundance in wet years, not quite significant. American shad, though, definitely had significantly more fish in wet years. Delta smelt, trend is not super clear, but striped bass and longfin smelt definitely have higher populations in wet and neutral years than droughts. Looking at the fish community-wide, we did a principal tensor analysis to see whether what groups we could group the fall midwater trawl catch into. And we found that 3 groups, threadfin shad catfish, a marine group, and a freshwater brackish group. We then looked to see how much the communities change with droughts, and we found a significant shift in community structure between droughts and wet periods for the threadfin shad catfish and for the fresh brackish cluster, but not for the marine cluster. They seem to be less influenced by outflow. So, as a whole, outflow really decreases with drought. This triggers an increase in salinity, also increase in Secchi and nitrate. Temperatures increase with drought. This causes an increase in microcystis and a decrease in a lot of our favorite fishes, as well as chlorophyll. Zooplankton and some of the other fish don't have quite as severe a change, and it can be variable with region. In 2021, we saw outflows even lower than past droughts. Salinity, temperature, Secchi were even higher than other droughts, making microcystis even higher than other droughts, though a little bit higher chlorophyll, which is nice. However, a lot of our pelagic fish continued their downward trends. Surprisingly, longfin smelt had a decent year, and salmon outmigration survival wasn't too bad. We won't really know what's going on with salmon until they return. And some of these factors we haven't had a chance to assess yet. So what does this mean for the future? Well, it's going to keep getting hotter and keep getting drier. We're going to have more frequent droughts as well as more frequent floods. We need to think about how we're managing the system. How do we optimize a limited amount of water for all of these conflicting species and human needs. We need new tools, and we need to plan for worst case scenarios earlier and more often. I don't know what all those tools are, and I totally know this is a very difficult question as to how to plan. Someone is always left feeling like they've been left out, whether that's a salmon or people who want water in the winter or people who want water in the summer. We need to be more creative about balancing those conflicting interests to the environment, people, and species. And with that, I will take any questions. Thank you very much.
Great, thank you so much, Rosemary. That was definitely, I could see a lot of work going into collating all of that data. So I really appreciate you taking the time to walk us through it. I'll check if there's any hands raised. And again, I probably didn't say this, but if you have any questions, you can raise your hand and I can turn off your mute so that you can ask your question. It looks like Barb has a question.
Hey, Rosie. I don't know much about Potamacorbula. That increase in grazing rate, is that on an individual level, like I, a single clam, eat more during droughts, or is it like there are more of me during droughts?
It's some of both, because the chief impact is there's more of me during droughts and they're moving further upstream, which means there's more of them in our sampling range. But it's also tied to temperature. So when the temperature is higher, which you get during droughts, an individual clam will graze more. Plus there's more clams equals higher grazing rate overall.
Excellent. I was like, I don't know much about the metabolism rate of clams, but just like striped bass, it goes up with temperature. Got it.
Looks like Deidre also has a question. Sorry, I was trying to unmute you.
Unmute. There we go. Thank you, Deirdre Desjardins. I'm wondering, so Peterson and Becerra's published a retrospective where they found the clams move up estuary and down estuary with salinity, as you found. But do you have any idea, once we have a severe drought like we've had in 2021, how long it takes them to move back down? Do you have any sense?
Yeah, so I initially looked at, outflow versus the position of clams, and I found that was not a great fit. But if I looked at the previous year's outflow versus position of clams, that was a much better fit. So there's about a year lag, and there's still a lot of noise in it. So it's probably there's probably some effects going on into 2 years, but they do have a pretty rapid life cycle. So I would guess that it would be, in the year to 2 year range for reversing that. But we haven't really dug into that yet.
Thanks. That sort of the memory of the system is sort of important for looking at these effects. Do you also, there's some research showing that reduced channel flows result in increased growth of macrophytic vegetation such as Egeria densa. Have you considered that at all?
Yeah, and we did look at invasive weeds, Egeria densa and others a little bit. The data are very noisy, and honestly, it looks like there's just been kind of a steady increase in weeds over the past 20 years, but we're missing a couple of years of data. However, like 2017, which was obviously a super high flow year, we didn't seem to have reduction of weeds, at least in a lot of the big open water areas like Frank's Tract in particular. It was as thick in 2017 as any of the drought years. So I don't know if we need multiple high flow years in a row, or there may be some differences in like the really high flow channels where Egeria might invade during droughts and leave during wetter years, whereas you don't see that in some of the flooded islands. That's something that we would like to dig into more. Unfortunately, we don't have as much of a long-term data set on the aquatic weeds as we do for some of the fish and macroinvertebrates.
Did you see, I mean, so the pod synthesis team found in 2010, they hypothesized that there was a regime shift that favored centrarchids and submerged vegetation and a shift in zooplankton. Is that too big a question for your analysis, or?
We didn't specifically look for regime shifts. There's certain techniques that we would have looked for in particular. We did see that, 2020, this current drought, the past 2 years, seems to be more intense than previous droughts. And that could be some of the regime shift going on. Also, just the time since the pod has had almost continuous drought. We've only had a couple of wet years, and we haven't had multiple wet years in a row. So it would not surprise me if the drought was kind of exacerbating that regime shift and keeping us stuck in that pod regime.
These are great questions. In the interest of time, I'm gonna have to stop, but I want to thank you, Rosemary, and I encourage you, if there's a lot of their hands up, I'd encourage you to reach out to Rosemary, because that was a lot of interesting data she shared. So thank you. So next we have James Gilbert, who is from UC Santa Cruz and NOAA's Southwest Fisheries Science Center.
All right, thank you, Miles. So today I'll be talking about temperature management in drought, moving a little bit farther upstream from the Delta. I'd first like to acknowledge my co-authors, Miles Daniels and Eric Danner at the University of California, Santa Cruz, and NOAA Fisheries, Southwest Fisheries Science Center, respectively. So specifically, I'll be talking about the role of different forecast input errors in projecting out and managing temperature in the Sacramento River, using drought as a critical case study. For a little bit of background, I'll talk about Sacramento River temperature management and how models and uncertainty fits in with that. And then talk about some analysis looking at reforecasting or hindcasting, looking at the 2021 drought as an instructive and informative means of understanding uncertainty. So for anybody that needs a refresher, the upper Sacramento River is host to several runs of salmon. I'll be talking about the endangered winter run Chinook salmon. They spawn in the upper reaches of the Sacramento River below Keswick Dam. I've indicated the approximate region there with that red oval on the map. They spawn at the sort of the region of the river right below Keswick Dam, and they spawn in the middle of the summer, which means that they are susceptible to mortality from warming temperatures in the river, which makes the operation of Shasta Dam and Reservoir for managing cold water and thermal habitat conditions an important consideration. So temperature-dependent mortality, or TDM, is an important measure or metric of this water temperature management. And over the last 20-some years, most years TDM has been quite low. The histogram on the left shows that the majority of years we have TDM less than 20%. But it's in these dry years, notably 14 and 15, and then last year, that we have these higher TDMs. And the plot on the right shows the TDM over the last 22 years plotted against May 1st Shasta storage. And we see that once Shasta storage gets below about 3 million acre feet, that TDM tends to increase quite quickly. So this really points to the effect of drought as it manifests in storage and its impact on salmon egg mortality. Just a quick overview of how temperature management in the process works. Normally there's an interagency discussion of hydrology and operations coupled with temperature modeling to project out what temperature conditions would be in the coming season. The models involved here provide projections across the season, but there's always uncertainty with these projections, and that uncertainty is viewed as an impediment to more certain planning and having better options. But this uncertainty is not very well characterized to date, which leads to the question, how do the uncertainties in the drivers of this system affect our outcomes of interest, in particular temperature and temperature-dependent mortality? So I mentioned models. The conventional approach is to have a reservoir model for Shasta that accounts for the vertical stratification and selective withdrawal at the dam face, a model for Keswick Reservoir, and a model for the river, both of those taking into account mixing and heat exchange, and then a model of the biological impacts, the consequences of temperature on egg mortality in the river. Each of these models takes a range of inputs, and for things like inflow hydrology and weather, these are themselves uncertain, especially in a forecast environment. I'd also add that things like operations and the spawning timing and location are also uncertain and complex in that they depend on the effects of inflow hydrology and weather as well. So how do we start to get a handle on all this uncertainty? The method I'll be talking about today is just simply revisiting forecasts as they would have been made in previous seasons and using the forecast information that was available, but combining that with actual forecast or actual reservoir operations. And because we're doing this in a modeling environment, we can combine and remove different sources of uncertainty and see what the important effects are. And like I said, I'm using 2021 as a initial case study. It was a drought year and a challenging year for temperature management. In this case, I'll be talking about results using a set of models for Shasta, Keswick, and the upper Sacramento River using the NIMS rapid models. These are simplified temperature models. They run at a daily scale and represent the reservoirs as one-dimensional models. The temperature-dependent egg mortality component is the NIMS TDM model. And in terms of the uncertainty of the inputs, I used the 90% and 99% exceedance Bulletin 120 forecasts from various months in 2021. I used a range of meteorology from 2013 to 2020 as an attempt at a representative variation in meteorology, and a composite of red distributions in the river, again, selected from some recent years. And this whole process is repeated multiple times on a monthly basis to kind of mimic how the temperature management and modeling process proceeds. So we start in February with the information available, information that was available in February of 2021, including forecasts and Shasta reservoir conditions, run it through the end of the season, and then advance to March, April, and May, etc. The first set of results here show the impact of considering hydrologic variability alone. And in each of the plots on the left, the black line is the observed or actual Shasta release temperature, and the shaded region is the results from the simulations. And in this case, that shaded area represents the difference between the 90% and 99% inflow forecast hydrology. We can see at the top that starting in February, the error in the hydrologic forecast and the uncertainty or the spread in that forecast has a big effect on the release temperature. And the error, because the forecast was wetter than it actually ended up being, shifted the whole release temperature colder. And the fact that the shaded area is wider indicated that there was a bigger effect of that input on the result. As we go down the plots, we see that the shaded area converges on that black line, and the uncertainty starts to diminish. And with the May forecast being very nearly the observed temperature and with a much lower uncertainty. When we look at the same thing, considering meteorological variability, we see a little bit different pattern. Again, February, the uncertainty has the biggest impact. But instead of really diminishing down to just kind of a line, that uncertainty kind of persists throughout the whole season. And the other thing I'll note too is that whereas the hydrologic forecast had a larger impact on the bias, that is the sort of average level of temperature compared to the observed, meteorology tends to just expand the uncertainty around a single answer. Kind of expanding on that point, we can look at what the absolute error in the input forecast has on the bias in the simulated result. This time looking at the average temperature bias over the July through September period for Shasta temperature releases. In this case, we see that for a hydrology forecast in the plot on the left, a forecast error volume of 2 to 3 million acre feet, or 2 to 3 million cubic meters has a much bigger impact in February and March than it does in, for example, April and May. Again, looking at the same thing for meteorology, we see that the overall impact is lower. The y-axis is actually scaled by a factor of 2 lower than the plot on the left, and that the effect is a little more consistent across the different forecast dates. Now, if we move downstream and propagate this temperature information from the Shasta release down through Keswick and into the river, we can calculate temperature-dependent mortality. And because each TDM estimate corresponds to one forecast date, I'll be showing a series of plots here, and this is just a quick introduction or orientation to that. The dashed line will show the final 2021 temperature-dependent mortality of 75%. This is just provided as a reference. And then the range of forecasts for a given date will be a vertical bar with that forecast initialization date corresponding to where it is on the x-axis. The location of that bar on the y-axis is a measure of its accuracy. And then the height of the bar is a kind of a rough measure of that uncertainty. So again, to start, I'll look at the results of just considering hydrologic input variability or uncertainty. And in this case, as we go from left to right, starting in February through May, we see that the uncertainty or the height of those bars diminishes through time. And we would expect that based on what we saw from the Shasta release temperature, that it got more certain and more correct through time. And in particular, we converge on a more accurate estimate of TDM by May. And this is, I think, a result of just the forecasts and the eventual operations slowly becoming more consistent. We can add on top of this, in this case, the impact of considering the historical red distributions versus what actually occurred in 2021. You'll see here that the lighter shaded portion of the bars only increases the top end of the TDM. And this is a result of the 2021, the actual red distributions being much more compressed compared to recent years. And then finally, we can add on the effect of meteorological variability. And in this case, it has the impact of mainly expanding TDM at the low end. This is, again, a result of using recent historical data that tended to have somewhat cooler, a somewhat cooler bias. You'll see here that even in February, we have a fairly wide range of TDM estimates, but it doesn't cover the full range. It does provide some information, as uncertain as it is. So I think there is at least some qualitative potential to using an approach like this for kind of an early warning indicator. Of course, this is just an initial analysis, and there's lots of things to do with ongoing work. One key issue here is that all the inputs are assumed to be equally likely, which is in some cases a pretty coarse assumption here. So one thing we can do is refine the quantification of uncertainty on the inputs, which would allow us to then translate the results into a kind of a probability distribution of TDM for each month. And that would lend itself very well to more of a risk-based projection and information for management throughout the early part of the season. So to wrap up here, drought manifests as low storage in reservoirs, and that in turn affects cold water and places constraints on temperature management. Understanding how we can take all the information available for projecting out options for temperature management, I think, is an important part and an important next step in improving the seasonal planning for temperature in the Sacramento River. The initial analysis we've done here indicates that despite all this cascade of inputs and uncertainty, there may be some potential for at least qualitative use of TDM risk early in the season, and then quantitative, more specific quantitative value after that kind of March to April transition. Hydrology is important to the extent that we can improve hydrology forecasts, especially in dry years. I think that could go a long way to improving TDM prediction, most notably in accuracy, with potential trade-off in uncertainty. And then meteorological effects seem to have a lower impact in terms of bias, but they still matter for variability around our predictions. Uncertainty is unavoidable, and so planning for precise targets in dry years is always going to be challenging. And again, I'd advocate for perhaps considering a more risk-based or probability-based approach, and looking broader scale into multi-year planning and impacts for drought. As previous speakers have mentioned, multi-year hydrology can be quite uncertain with extremes in magnitude and interannual variability. So I think there's lots of important and exciting work yet to be done. And with that, I'm happy to take any questions.
Great, thank you, James. That was a really nice example of how physical drought impacts correlates with biological impacts. So we have a few minutes for questions if anybody has any. So I'm asking Barb, can you, sorry.
Can you hear me, Miles?
We can hear you. That was my mix-up.
Oh.
Sorry, Barb. We can hear you. Sorry.
James, super interesting talk, sort of trends in the way I'd sort of expect. And one question I have is, do you think that the sort of the sensitivity that you showed in hydrology compared to, say, temperature, is that mostly driven, would you say, by the fact that, at least in the case study year that you chose, there was simply that temperature ended up being close to what we thought it was? So I'm kind of asking sort of, is the mechanism underlying the hydrology change more important, or is the sensitivity coming from simply there was a bigger change in the reality compared to February expectation in the hydrology data compared to the MET data?
I think, if I'm understanding your question, I think in particular for 2021, the change in the forecasts between the months and the total volume by which the forecast in February, for example, was off from what ended up happening, was particularly important in shifting the results around. we want to expand this to look at other drought years too, to see if the hydrology forecasts have a similar magnitude of effect. But I think, again, if I'm understanding your question right, I think that's correct, that just the sheer magnitude of the impact of hydrology when the reservoir started kind of in a, this very sensitive storage level, that has the biggest impact.
All right.
Thank you.
Well, great. Yeah, in the interest of time, sorry to cut other people off. I know there's other questions. I apologize, but I would encourage reaching out to James for follow-up questions. Okay, so next we have Susanne Brander, who is an assistant professor at Oregon State University, and I'll invite her to share her slides as well. Thank you, James.
All right, can you see my screen?
Yes, we can.
All right, wonderful. Great. Thank you so much for the invitation to give a talk today. Today I'll be talking mainly about the influence of salinity on pyrethroid toxicity in a model estuarine fish, Menidia beryllina, but we'll be comparing this to some results from the same grant in Delta smelt. I'd like to give all of the credit on the work I'll present today to PhD student Sarah J. Hutton and postdocs Samreen Siddiqui, Amelie Sagara, as well as lab technician Emily Peterson. And all of this work was funded by the Delta Stewardship Council over the past couple of years. And so we've heard a lot about drought and downstream effects from others in this session. And of course, another consideration is how organisms may be affected by changing salinities in the context of pollutant exposure. And we know also that the San Francisco Bay may experience up to a 9 PSU increase in salinity by 2090, considering some modeling projections. And we also know from research done over the past decade or so that salinity can actually alter and sometimes increase the bioavailability of some chemicals, especially those that tend to be more hydrophobic, which include pyrethroid pesticides, which are heavily used in the San Francisco Bay. And empirical work done under this grant shows that pyrethroids have an increased fraction in water as salinity increases. As you can see on the bar chart shown here across a number of different pyrethroids. This may be due to a couple of different factors, including potentially binding to salts, as increased salinity causes chemicals to bind to particulates in the water. This would happen with organic matter in the field as well, for example. So there's really a need to not only understand the way organisms are responding potentially differently to chemicals as climate change progresses and we have factors such as increased salinity, but we also need to understand what's happening with the chemicals themselves. So pyrethroids, as many of you know, are synthetic insecticides. They're commonly found in the sediment and surface waters and estuaries and bay regions. This is in California, but now these are commonly used nationwide and even globally. Pyrethroids are far more toxic to fish than they are to mammals. They induce neurotoxicity by acting upon sodium channels in the brain. They can also cause unintended effects. So the neurotoxicity is an intended effect of pyrethroid pesticides, and it's how it causes toxicity and death in insects. But it can also cause decreases in reproduction, altered sex ratios, as well as behavioral changes in fish. And an example is that it has been detected in Cache Creek, which is an important spawning area for delta smelt at levels up to 150 nanograms per liter. And effects are seen even below 1 nanogram per liter in the laboratory. So why study behavior? Well, first of all, given that pyrethroids are designed to be neurotoxicants, it's not surprising that they would have an effect on behavior and that this would be a relevant endpoint. So we know that this is a sensitive endpoint, that we have high confidence that we will detect an effect, and we've already seen that early life exposure to pyrethroids does cause behavioral impacts, and it's ecologically relevant. Neurological effects can influence the way a fish feeds, whether it's able to avoid being predated upon, or whether it's able to obtain prey itself or to find a mate. And so we exposed Menidia beryllina to 6 different pyrethroids. They were exposed at 5 days post-fertilization, so they hatched out into these treatments. And this was a short-term exposure for 96 hours or 4 days. And they hatched out about 1 to 2 days into that exposure. And following those treatments, larvae underwent a 40-minute behavioral assay with 3 10-minute dark periods interspersed by 2 5-minute periods in the light. And this is modeled after other behavioral assays that have been conducted in zebrafish, silversides, and delta smelt across the Brander and Conan laboratory at UC Davis. And so we used an instrument called the Noldist EthoVision to do these behavioral assessments. This is a video tracking system that is used to auto-record animal activity and movement. And it can measure small changes in a lot of different parameters, including distance moved and time spent in a certain area of the well. And again, these fish are just a few days old and a few millimeters in length. And so to get an idea of what this looks like, I've provided a short video, and you can probably see these tiny little black dots moving around in these wells that we've actually designed from glass beakers, so we're not worried about pyrethroids sticking to plastic or the chemicals in plastic interfering. And so all of our observations were recorded in this way and then analyzed following recordings. And that's just showing you where the camera is on top there. It's able to record very small changes in behavior. And so getting to results, and here I'm just showing you a snapshot of the results. We'll be submitting this for publication soon. And you can see that from this heat map that behavioral responses were complex. What this is showing you is a heat map with normalized behavioral responses. They're all normalized to controls. Purple represents hypoactivity, so lower activity or lower distance moved in this case. Orange represents hyperactivity, or a larger distance moved. And you can see that with permethrin, for example, the most effects were observed at this middle concentration of the chemical and at the highest salinity. And which salinity caused the greatest impact varied depending upon the pyrethroid. So from this research, we're still able to say that pyrethroids affect behavior at low concentrations, at concentrations detected in the environment, and we're able to say that salinity has an influence, but there's not necessarily a golden rule of how salinity affects pyrethroid toxicity, and that's something we still need to do a much deeper dive into. But overall, the higher 6 PSU salinity had fewer changes in total distance moved relative to the 2 lower salinities that we tested. So moving on to something called thigmotaxis, which might be a word you haven't heard before. What thigmotaxis refers to is when you're talking about an assay conducted in wells to evaluate fish behavior, you're talking about the time spent near the wall. So more thigmotaxis means more time spent near the wall, less means more time spent in the center of the well. More can indicate anxiety or avoiding kind of the center area, the center of that arena. Being in the center indicates increased boldness. And what we saw was that anxiety appears to decrease with salinity for cypermethrin, but that we saw the opposite effect for permethrin. And so just because the names of those pyrethroids sound similar doesn't necessarily mean they're going to cause the same effect. And I'll talk a little bit more about why that is in a couple of slides. So again, we saw that in some cases salinity did have a strong influence. In other cases it didn't. Across the board, we saw a myriad of responses at varying concentrations, most of which were concentrations detected in the environment. So comparing this back to delta smelt, and this is work that was published last year in 2021, we saw that anxiety indicating responses like thigmotaxis and delta smelt are also differentially impacted by pyrethroids and are dependent on salinity, on the same range of salinities. But we see that there are differences, that bifenthrin, for example, is more toxic than permethrin. And what these radar plots are showing over here is that at 6 PSU, you see this anti-thigmotaxis or increase in boldness by those delta smelt, which might be increasing their risk of being predated upon, depending on salinity. Of course, you see that there's very little response at 0.5, but a high response at 6. So that increased salinity that is forecasted to happen in the delta may make some of these responses stronger and may increase the ecological risk of pyrethroids to endangered species like delta smelt. There are others that have found similar results. So there was a paper out in 2019 that saw similar responses in zebrafish, that exposure to permethrin might decrease anxiety-like behavior in the zebrafish, basically altering their preference for, being in the weeds, hiding, rather than being out in the open. So this gets a little bit more into the weeds, so to speak, but we also looked at the influence of how hydrophobic or how low the water solubility of a particular pyrethroid is on those anxiety levels. And what's listed here on the side of the slide, it's something called an octanol water partition coefficient, and it's a measure of how hydrophobic a chemical is, just a way of quantifying that. And basically what we saw was that at a lower salinity, that anxiety-like behavior seems to increase with increasing hydrophobicity. And at the higher salinity, we see that anxiety-like behavior decreases with increasing hydrophobicity. So you see an opposite effect or opposite impact on that behavior, depending on whether the salinity is higher or lower, and also depending on how hydrophobic the particular pyrethroid is. There also seems to be an effect of the type of pyrethroid, and there are generally two types of pyrethroids. Type 1 lacks a cyanide functional group, type 2 has one, and their name typically starts with CY to indicate that. And we also see some variance in the influence of behavior dependent upon whether the pyrethroid is classified as a type 1 or a type 2. So lots of things to dig into here. But before I finish today, I did want to talk a little bit about some new results from a related multi-generational study that we're conducting under the same grant. And so for this study, and this was based on studies that have been done in bifenthrin, where both multi and transgenerational effects of early life exposure have been demonstrated. F0 embryos were exposed just for a short time period, for that 4 to 5 day time period, similar to what I discussed earlier for the behavioral assessment. And then they were reared in clean water for 8 months, so long experiment. Fingers crossed, most of them survived, luckily. They were spawned, and then the F1 larvae, which were indirectly exposed as germ cells within their parents, their behavior was evaluated, and then we're also evaluating other endpoints such as development, hatching rate, and looking at things like sex ratio and gonad size in their parents. And what we found was really interesting. In the F0, we saw, in terms of behavior, a lot of hypoactivity when looking at total distance moved. And we saw this at both salinities, but the effect was a bit stronger at the 10 PSU salinity in comparison to the 6. And then when we looked at their offspring, we really saw fewer effects overall on behavior, but in some cases there was an increase in hyperactivity in comparison to the parents. So we may be seeing this kind of compensatory response that has been seen with other factors in terms of multi-generational assays. And things that are still in progress with this work, which took a few more months than anticipated. We see that preliminary results suggest that bifenthrin caused delayed hatching at 6 PSU, and cyhalothrin caused a similar effect at 10 PSU. And then we also observed more developmental defects in these treatments compared to the others. And so again, these were in fish that were not exposed directly. Their parents were exposed for 4 to 5 days in early life reared for 8 months in clean water, and these are fish that were spawned from those adults only exposed in early life. So this was a short-term exposure that had longer-term impacts. Also in the F0 adults, we found that some of the males had lower gonadal somatic index, so smaller gonads, compared to their 10 PSU counterparts in some of the pyrethroid treatments, including bifenthrin. So a lot of results. What does this all mean? Well, we saw that salinity causes differential behavioral responses in urohaline fish exposed to environmentally relevant concentrations of pesticides. We saw this in silversides. We also saw effects in delta smelt. They aren't necessarily as sensitive to the same pyrethroids, but the overall story is that pyrethroids do cause behavioral impacts that could affect things like ability to obtain prey or the chances of being predated upon in the wild. We see that we see both behavioral and endocrine-related effects in indirectly exposed offspring. This isn't the first time these effects have been documented for bifenthrin, although in this case, the window of exposure was much shorter than it had been for previously published work, where the fish were exposed for a couple of months rather than just a few days. But even when they're just exposed for a few days, we do see these kind of carryover effects in the F1 offspring. And as I mentioned earlier in the presentation, the San Francisco Bay is expected to experience likely an increase in salinity. We don't necessarily see that all of these effects are aligning with things being more severe at higher salinities, but we see that varied salinities really need to be considered when we're thinking about what the impacts of pollutants might be, both in the immediate time frame as well as over more of an extended time frame, because we are seeing immediate effects on things like behavior, which makes sense when you're considering something that is a neurotoxicant, but the latent effects we're observing are on fecundity and other endocrine-related endpoints. And we are currently analyzing gene expression with a focus on both osmoregulation and endocrine function. So there should be even more data to look at soon. Thank you so much for your time. And if there's time for questions, I'm happy to take some.
Great, thank you, Susanne. That was some really exciting lab that you guys are doing. And we do have time for questions. And I'll just remind people they can raise their hands, or I've been trying to look at the Q&A box as often, but there's too many windows open.
Sure. And pictured here are the people who did all of the hard work for all of these experiments. So, and our contact information if you have questions.
It looks like we have one question in the Q&A box from Barb. Would you mind stating that question, Barb? If not,
I can read it if that's easier.
Sure, yeah.
Barb says, have you done any treatments on pollutant combos? Interested in how effects may be amplified, or maybe, as in the differential thigmotaxis responses canceled out? It's a really excellent question. And the answer is I would love to look at effects of mixtures and make this even more realistic. The amount of work involved in exposing organisms to 6 different pyrethroids at 3 different salinities, and also doing the multi-generational work meant that that wasn't possible for this particular grant. But I do feel like that is the next, that represents the next question. And part of the challenge is that pesticides are regulated as if animals are exposed to them singly rather than in combination. And so if we are trying to determine thresholds, regulatory thresholds, which was part of the objective of this grant, to determine whether the thresholds that have already been determined as safe for the bay are low enough, we did need to look at them individually. But I do agree that it would be really interesting to look at them together. I don't know that we would see that the effects cancel each other out, given, especially in terms of neurotoxicity, but we might see some interesting effects of exposure to mixtures for some of the longer term impacts, the endocrine related impacts, and some of the pyrethroids tend to act as estrogens and others tend to act more as anti-estrogens or androgens. So really good question.
Great. Well, thank you so much. I really liked how you were able to kind of address in the laboratory some of these questions that are occurring out in the field, and things that you can't control in the field, at least you can control in the lab. So that was really interesting to see.
Thank you.
Yeah. All right. So our last but not least presenter today is Flora Cordoleani. She's a project scientist at UCSC, as well as NOAA's Southwest Fisheries Science Center. And so Flora, you can share your screen now. Thank you.
Okay, can you see that?
Yes, we can.
And can you hear me?
Yes, we can.
I'm assuming. Yes. Okay, thank you, Miles, for inviting me to this session. I'm really happy to talk about our favorite fish, like Rosemary said, Chinook salmon in the Central Valley and drought effects. And especially, I'm going to talk to you about a project where we used otoliths or earbones to look at the success of spring run Chinook salmon population in the California Central Valley volatile and warming climate. And this work has been done in collaboration with Corey Phillips from Metropolitan Water District, Anna Sturrock from who is now at the University of Essex, Alyssa Fitzgerald from UCSC and NOAA Fisheries, and Rachel Johnson from NOAA Fisheries. So as you all know, the California Central Valley is known to be a highly volatile, variable, and extreme environment that has been historically marked by cycles of flood and droughts. And Chinook salmon population have evolved to adapt to such a variable environment. However, in addition to those natural events, the Central Valley rivers have been heavily modified due to human activities, such as mining, dam implementation, and other water diversion projects. And this has led to a simplification of the Central Valley watersheds, and especially to the loss of 95% of the floodplain habitat, as well as an important loss of high elevation habitat that are now permanently blocked by impassable dams. And this has taken a toll on Chinook salmon population, and especially on spring run Chinook salmon in the Central Valley. They were one of the most abundant run of Chinook in the California Central Valley. And as you can see on the figure on the left that shows their historical distribution, they were found in every major stream and rivers where high elevation habitat was accessible. They are now listed as threatened under the Federal Endangered Species Act, and you can see their current distribution on the right. They have been completely extirpated from the San Joaquin Basin, even though now there is a restoration project, reintroduction program, so hopefully those fish will get back into this basin. But there is only what is called self-sustaining population. There are only 3 of them left, which are highlighted in yellow here on the map, and are Mill, Deer, and Butte Creek. There is also a hatchery in the Pedro River and some other dependent population, but for today, I'm going to focus on the 2 uppermost stream in this map, which are Mill and Deer Creek. So spring run are characterized by a large phenotypic plasticity that allows them to buffer against disturbances from the environment or human perturbations. And this plasticity is expressed through various life history strategies at multiple stages of their life. And especially, they are known to express different juvenile rearing and migratory strategy during their freshwater phase. And you can see that on the figure on the bottom right that shows the size of the fish living Mill and Deer Creek during the different months, so from November to June. And I highlighted in 3 different colors the 3 main groups of the 3 main strategy that we observed in this population. So we have in red the fry, that are small fish living tidal tributaries soon after emergence, the par or smolt rearing strategy that live during the spring at larger sizes. And then the last strategy is what we call the yearling component, which is a very unique life history strategy that is not much observed. It's pretty rare in the Central Valley. And that the strategy, those fish stay an entire year in the Null reaches before migrating to the ocean the following fall or winter at larger sizes. So the question we had is, how can this phenotypic diversity help them cope with the California volatile and future warming climates? And especially we were interested in knowing which life history strategy is more represented in the adult returning to spawn, and how do those successful rearing strategy vary among the different hydrological conditions that juveniles were experiencing during their freshwater phase. And so for that, we used otoliths, as shown on the figure on the bottom left, which are ear bones that were collected from carcasses by the California Department of Fish and Wildlife during their annual carcass surveys that they conducted in both Mill and Deer Creek for several years. And otoliths are like trees, so they grow ring, as you can see on this little figure here. And so you can estimate juvenile growth by looking at the distance between two consecutive rings or increments. And you can also estimate the number of days that juveniles spend in different watersheds. And it is also an incredible tool because it allows to record the chemistry of the water in which the fish were spending time. And so we used otoliths to do a movement reconstruction through a strontium isotope analysis that was performed at UC Davis. Because in the Central Valley, the 87-86 strontium ratio, especially, the values of this ratio are pretty specific to each watershed. And so by looking at this difference in strontium ratio throughout the otolith of a fish, you can reconstruct the rearing and migratory behavior of each fish. So as I mentioned, I'm going to focus on Mill and Deer Creek watershed, which are in the upper part of this figure here. I combine Mill and Deer Creek otoliths because, as you can see, they are pretty close geographically, and they're also very close genetically. And I want to mention that, as you can see here, there are no impassable dams in Mill and Deer Creek, so springmen still have access to high elevation habitats in the Lassie National Forests. So we were able to analyze about 123 otoliths across 6 different years from 2007 to 2018. And here the return years show the year the adult returned to spawn. So I'm going to show you a series of 87-86 strontium profile. And so let me run through this figure so you can understand it. On the y-axis, you have the 86-87 strontium ratio values, and those values are specific to different watersheds that I've shown on this map, on this figure. And then on the x-axis, you have the otolith radius, which is a proxy for fish size. And so for the first part of the profile, from about 0 to 200 micrometer, that corresponds to the incubation phase of the fish. So what we're really interested in is after about 200 micrometer, after their emergence, so all their rearing phase until they get to the ocean. So as you can see here, and then on the map on the right, you will see the movement of fish during their throughout their life in the freshwater. So you can see that the first series of profiles that we found were corresponding to the fry migration strategy, where fish stayed a very short amount of time in the Needle tributary, migrating to downstream habitats such as the upper Sacramento River to rear for a few months before migrating as small-sized fish to the ocean. We also found a series of profiles that corresponded to the smolt rearing strategy. So fish stayed a few months in the Needle tributary before migrating in the spring to the ocean. And then finally, we found another group of profiles that corresponded to the yearling migratory strategy, where you can see a longer rearing time period in Mill and Deer Creek tributary. So they stayed over summer, and then they grew and migrated to the ocean in the following fall or winter as yearling sites. So we found that the 3 migratory strategies that were observed in the juvenile trapping data were also observed in the adults that successfully came back to spawn. So we wanted also to see what proportion of each life history strategy was observed for each given return year. So on the figure on the left, I'm showing you the size distribution at natal exits, so leaving Mill and Deer Creek, for each return year from 2007 to 2018. And in parentheses, you see 2 immigration years because we assume that adult spring run return to spawn at age 3 and 4. So those correspond to the hydrological, to the years where the juvenile were in the freshwater and immigrated from the freshwater. And then on the figure on the right, you can see the proportion of each migratory or rearing strategy that were found in the spawning pool. And late corresponding to yielding, intermediate to smolt, and early to fry strategy. And so what we can observe is that for 3 of the return year, 2012, 14, and 18, which corresponded to dry and warm immigration conditions, the late migrating or the yielding strategy was dominant among the successful spawner. However, for the 3 other return years, 2007, 8, and 13, which were associated with wetter and cooler outmigration conditions, we had a mix of the 3 strategies found in the spawners. Okay, so finally, we wanted to explore how this life history strategy were going to be impacted by future warming temperature in the Central Valley. And Alyssa Fitzgerald did some very elegant temperature mapping that I'm showing you on these 2 figures. So we looked at the month of August, especially because we considered that the yielding strategy, which requires fish to rear over the summer, was going to be likely to be the most vulnerable to future warming climates. So we looked at temperature in the watershed during the month of August. And we looked at current condition, which is the left figure from 2005 to 2015. And then she did some prediction for the year 2080. And I'm showing you all the stream of the Central Valley where spring run were either historically or currently found. And we consider that a stream is thermally suitable, a reach is thermally suitable if the temperature stays below 15 degrees Celsius. So in blue, you have the reach that are suitable and accessible to spring run. In yellow, you have the reach that were thermally suitable but are currently inaccessible either due to impassable dam or natural barriers. And then in gray, you have all the reaches that are unsuitable. So if we start with current condition, we can see that suitable habitat that is accessible is found in the upper Battle Creek, Mill, and Deer Creek watershed, as well as below the dams in Clear Creek, the Yuba River, and the Stanislaus River, and a little bit in the upper Sacramento River. If you look at inaccessible but suitable habitats, you can find some habitat in the north, so above Shasta Dam and Shasta Reservoir, in the northern fork of the Feather River, in the northern Yuba, in the American River Forks, and then a little bit above dams in Stanislaus and the Tuolumne River. As expected, if we look at this amount of suitable habitats for temperature in 2080, this both suitable habitat and accessible and inaccessible reaches shrunk. And we actually estimated that the amount of accessible habitat that was suitable would be divided by more than 2 by 2080. And this amount could actually be tripled if we were to provide access to those reaches that are currently inaccessible. So to conclude, we found that the Mill and Deer Creek yielding strategy acts as an insurance policy against droughts. During those warm and dry outmigration conditions, an entire cohort of Mill and or Deer Creek fish could have been lost without this yielding component. And we also saw that the other 2 life history strategies were also important during weather conditions. So because each of these rearing strategy is associated with different time windows and might require different environmental conditions at a specific time, restoring the mosaic of habitat that was historically found in the Central Valley watershed that could support those various life history strategy might be important for the persistence of spring rain in the future of California. And if we consider particularly the yielding strategy again, we've shown that the strategy needs cold water refugia to rear over summer, and that the amount of cold water pools is going to shrink with the future warming temperatures, especially below migration barriers. And so providing access to cold water refugia above those impassable dams would be critical for the success of spring rain population in the future climate, and especially will help preventing local extinction during those future drought cycles that are going to happen. And with that, I would like to thank all the funders and the partners, agencies that made this project possible. And I would like to thank you for listening to this last talk of the session. And I wanted to mention that this work has been published recently in Nature Climate Change. I'm showing here the reference if you're interested in the paper, and you can shoot me an email if you want to have access to this paper, or if you have any questions, and I will take any question as well.
Great, thank you so much, Flora. That was really interesting. So it looks like Steve has a question. You should be able to talk now.
Yeah, hi, Flora. Awesome talk. This is some really interesting data. One thing I'm wondering is what in terms of the temperature criteria, it looked like there wasn't very much accessible habitat even above the dams. And so being that if that was the most abundant historic run, I'm wondering what the temperature criteria is that was used to determine what's suitable.
So temperature needed to remain below 15 degrees Celsius to be suitable, but remember that's just for the month of August. So we did some temperature mapping for the spring, and you have more suitable habitat earlier in the spring. And again, also remember those are for 2005, 2015, and then compared to 2080. So it's possible that in the past there were even more. It's likely that in the past there were even more suitable habitat above those dams, since those temperatures might have been even colder.
Yeah, one thing actually, I think David Boughton from NIMS actually just published a really cool paper on habitat above the Merced and Tuolumne rim dams for spring run that might be interesting to check out, because it seemed like a lot of the conditions, the modern conditions, were actually pretty similar to historical. So maybe something just to check out. That was a really cool paper they just published as well. But thank you.
Yeah, I didn't know if it was published actually, so thank you. I will read this paper. And I was really interested to see that there is actually some suitable habitat in Stanislaus River. So even below the dam, there is still a little bit of habitat that could be good for spring run. So I'm really interested in hearing what's gonna happen in the San Joaquin Basin.
Now we just need to actually sample for those yearlings.
Yes, I've heard about that as well. That would be good if they take some tissue samples. Thank you.
We have a few minutes left. Looks like there might be something in the Q&A. I will read it this time to be nicer to the speaker. So Ted Sumner said, I was surprised that the Feather River never had suitable temperatures before or after climate change. That seems questionable for the low flow channel. Or there's some flexibility in keeping this reach cold.
Yeah, I'm not sure about the details. There seems to be, yes, I'm not sure why there is no water, no reaches below the dam that was suitable for Feather River. I have to say that at least I wasn't able to map, I think, or to map reservoirs in general. So it was really strictly constrained to main streams. So maybe that might have an impact if you had the forebay or impacts from other reservoirs on temperature in the Feather River.
All right, one thing I just wanted to say, I think a common theme between all these talks, which were excellent, were that, yes, there seems to be climate change drought impacts, but maybe there's ways around that. And one of them definitely seems to be planning, monitoring for it, and modeling. So that's at least what I saw as a common thread to all these talks, which again were excellent. I really appreciate everybody taking the time to come here and give them. And I think if there's no other questions, which I'm not seeing any right now, we are about the end of our time. And I do know that I think in 10 minutes there are lightning talks as well on this same Zoom link.
Great, thank you, Miles. Yes, we're going to take a 10-minute break. So everybody be back here at 10:50. And for the speakers that are coming up in the lightning talk section, please make sure you come on at the panelist level. If you can't reach yourself out of the audience, raise your hand and we'll elevate you up. Thank you.